All right, today we are diving into a technique that, and I'm not exaggerating here, completely changed the game in molecular biology. It was invented way back in 1975 by Sir Edwin Southern, and it gave scientists, for the very first time, a solid way to find one specific gene in an absolute sea of DNA. This is the story of the Southern blot, basically the original search engine for our genes.
Okay, so put yourself in a scientist's shoes for a second. Imagine an organism's entire genetic code, its genome, is like a massive library with millions and millions of books. Now, your job is to find one sentence on one page in one of those books. How on earth do you do that? It was a huge problem. They had the whole library, but no index, no search bar, no way to find that one little piece they needed to study.
So, what was the solution? Well, it was this really elegant technique called the Southern blot. And that search engine analogy, it's perfect. This method lets scientists cut right through all the genetic noise and home in on the exact DNA sequence they're looking for. It was a total game changer. Now, the central idea here is something called blotting. In a lab, blotting is actually a pretty simple concept. You're just transferring biological stuff, like our DNA, from a fragile jello-like gel onto a much tougher, more durable surface, usually a nylon membrane. The reason you do this is to lock the sample in place, which, makes it way easier to work with. And here's a fun little piece of trivia about the name. The Southern blot is named after its inventor, Edwin Southern, and it's specifically for DNA. Well, when other scientists developed similar techniques for different molecules, they decided to have a little fun with it, sort of a science joke. So the technique for RNA, they called it a Northern blot, and the one for proteins, you guessed it, a Western blot. Okay, so how does this genetic search engine actually work? Well, it all starts with getting that massive DNA library ready for the search, and that involves a few really key steps to cut, sort, and separate everything. First things first, scientists have to get the DNA out of the cells. Simple enough. Then they use these amazing little proteins called restriction enzymes. Think of them as tiny molecular scissors. They chop up the super long strands of DNA into millions of smaller, more manageable pieces. And finally, all those pieces get loaded into a gel and an electric current is run through it. This is called gel electrophoresis, and it sorts the fragments by size, with the little guys zipping through the gel much faster and further than the big ones. So at this point, what we have is basically a smear of millions of DNA fragments, all sorted by size. But our gene, it's still completely invisible, totally lost in that crowd. The next set of steps, well, this is where the real magic happens. This is how we make our target pop. And this is where it all comes together. First up is the blotting step we talked about, getting the DNA from that flimsy gel onto a sturdy membrane. Then comes the most critical part. Hybridization. This is where a special probe is washed over the membrane, and it's designed to hunt down and stick only to our gene of interest. And the final step? Detection. We just have to see where that probe ended up, and boom, we found our gene. So that probe is everything. It's our search term. It's a tiny, custom-made piece of single-stranded DNA that's basically a mirror image of the gene we're looking for. And because of how DNA pairs up, it's going to ignore the millions of other fragments and bind only to its perfect partner. And here's the clever bit. The probe has a label attached to it, kind of like a tiny glowing flag that lets us see exactly where it is. And there are all kinds of cool ways to flag that probe. The old school way was to use a radioactive label. You then put a piece of X-ray film on top and it would leave a dark band right where your gene was. But today, we've got safer and honestly more varied options. The probe can be attached to something that creates a colored spot, or a molecule that literally glows under a specific light, or even one that triggers a chemical reaction that gives off its own light. Pretty cool, right? Now, I know this all sounds like a super complex lab procedure, and it is, but its invention unlocked an absolutely staggering number of real-world applications that have changed everything from medicine to law enforcement and even our basic understanding of life itself. For decades, Southern blotting was a massive deal in genetic diagnostics. It meant doctors could do prenatal diagnosis for single-gene disorders like sickle cell anemia. They could literally look for the specific disease-causing version of a gene in a person's DNA. This was huge for families. It also became the technique that DNA fingerprinting was built on. By using probes that targeted these unique repeating parts of our DNA, they're called VNTRs, forensic scientists could create a pattern of bands that's unique to each one of us. This completely revolutionized crime scene investigations and paternity tests. And before we had the technology to just sequence entire genomes super fast, we had to map them first. Southern blotting was essential for those early mapping projects, especially for a method called RFLP analysis. It was all about looking at the different fragment sizes made by those molecular scissors we talked about earlier. And of course, for the researchers in the lab, its main job was exactly what it was designed for, sifting through huge genomic libraries to find and pull out one single gene they were interested in. This let them clone it, study what it does, and figure out its role in our health. It's really not an overstatement. And yeah, while newer techniques like PCR are often a lot faster for some things now, the fundamental principles of the Southern blot are still a cornerstone of modern genetics. It literally taught us how to search our own instruction manual. So just think about that for a minute. A technique from 1975 gave us the power to search our own DNA, and it changed the world. It really makes you wonder, as technology keeps moving at this incredible pace, what's going to be the next big search engine for understanding the amazing complexity of our own biology?